Hello, my name is Dr. Roderick L. Roll. Today, we will be talking about controlling the cell. To control the cell utilizes many cellular surveillance signals, both from an internal standpoint and an external standpoint. What controls cell division? There's four things we will talk about. The first one is called cell surface to volume ratio. Once the cell reaches a set ratio, it will divide. So let's look at these these two cells, cell A and cell B. Cell B is very large. So we're looking at the surface to volume ratio of cell B versus cell A. You do not need to remember the number, but just know that as the cell enlarges, this cell surface to volume ratio will decrease. So once the cell reaches this set threshold of 6, then it will divide. So if the cell wants to take up the same amount of space as cell B, it will just replicate until it has enough cells to make up that difference. So bigger is not always better. So the bigger the cell, the smaller the ratio. This is going to make diffusion of these molecules into the cell, the collision of these molecules into the enzymes, it's going to make all of that less efficient. Therefore, the cell will not be able to produce enough energy to sustain life. So bigger is not better. Smaller is better in reference to the cells. The second control mechanism is internal signals. We're going to discuss two of them: chromosome attachment and cyclins and cyclin-dependent kinases called CDKs. The cell has three important checkpoints: G1, G2, and M phase. The G1 checkpoint seems to be the most important one for most cells. If a cell receives a go-ahead signal at G1 checkpoint, it will usually complete S, G2, M phase, and divide. So the checkpoint is a barricade, is a roadblock, is a beautiful big wall. If the cell does not receive the go-ahead signal at the G1 checkpoint, the cell will exit the cycle. The cycle is the cell cycle. It consists of interphase and M phase. If the big beautiful wall is not destroyed, is not lowered, then the cell cannot proceed through the cell cycle. So it will go into a non-dividing state called G sub 0. An example of such a cell is a nerve cell, a liver cell, or a muscle cell. Humans have 46 chromosomes. So 2A is still talking about an internal signal. So without full chromosome attachment to the spindle fibers in late prophase, the cycle stops at the M checkpoint. So again, we have this big beautiful wall. This big beautiful wall needs to be destroyed or lowered. This wall is chromosome attachment. So the centrosomes created the spindle fibers. These spindle fibers are attached to the kinetic core within the centromere. If one of them does not attach appropriately, then the wall stays up. If all of them attach, then the wall can come down. When the wall comes down, the cell will proceed through the M checkpoint. So with full chromosome attachment, all 46 chromosomes have spindle fibers attached. The big wall comes down, green light, go. The cell will continue to go through the cell cycle. If one of those spindle fibers is not attached to the chromosome, an error will occur. Therefore, you do not want to reproduce that cell. You need to fix it. So the cell can fix itself or it can destroy itself through a process called apoptosis. So this is an internal signal. This internal signal assures that daughter cells have the correct number of chromosomes. Each daughter cell must have 46 chromosomes. The second internal signal is cyclin-dependent kinases and cyclins. When these two entities come together, we will create a MPF complex. So in this image to the right, we have a green shape and a red shape. The cyclin is the green shape, and the red one is cyclin-dependent kinases, better known as CDKs. When those two entities come together, that is the go-ahead signal for the cell to continue through the cell division or the cell cycle. So the MPF complex must be created. This is an internal checkpoint. It typically pushes the cell through the G2 checkpoint. It typically lowers that big beautiful wall. So in the presence of MPF, the cell will continue. So let's look at this image. What happens is we have a set number of cyclin-dependent kinases. That is the gold brick in this image. Let's say we have 100 of those molecules within a cell. The purple cyclins must be produced. They are a protein. So if we have 100 CDKs, we need to create 100 cyclins. So at first, as we go through G1, we might have 10 present. As we get to S phase, we might have 25 to 50. As we get to G2, we might have 75, 85, 100. Once we have created enough cyclins, they will bind to the CDKs and create the MPF complex. This is going to lower the big beautiful wall that is holding the cell back from proceeding to the M phase. This was observed in frog eggs. So look at this chart and you will see that cyclin molecule, the purple line, is increasing. Once it gets to a set threshold, it will complete the MPF complex. This again will push the cell through the M checkpoint. Other cyclins and cyclin-dependent kinases give the go-ahead at a G1 checkpoint as well as G2 checkpoints. So it can serve as many different checkpoints. Scientists have so far discovered that we have 3 types of cyclin-dependent kinases and several different cyclins, all of which will complete a MPF complex. The third thing that pushes the cell through cellular division are chemicals. Chemicals such as hormones or growth factors. Growth factors are external factors that are released by certain cells that stimulate other cells to divide. The classical example is with getting cut. Once you are cut or you have a laceration, you're going to bleed. You're going to release platelets. These platelets are going to generate a platelet-derived growth factor. This is a hormone that is released by this cell type, but it affects a different cell type. So the definition of a hormone is a chemical that is produced by one cell type that does not affect that cell type. It affects a different cell. So the platelet-derived growth factor is a hormone that is going to be released, and it is going to affect cells such as fibroblasts. These fibroblasts are going to go through cellular division to repair the wound. So when an injury occurs, the platelet will release PDGF into the vicinity. This is going to cause proliferation of the fibroblasts, which will help the wound. This slide shows where the fibroblast is. It is deep within the connective tissue that resides in the dermis of the skin. So again, when a laceration occurs, an external factor called a growth hormone is going to be released from the platelets that will stimulate the fibroblast to repair the laceration. Based on the Campbell book, the 10th edition, 50 different growth factors have been discovered that can trigger cells to divide. This is a list of some of those growth factors. This is a few more of those growth factors. The fourth way to control the cell is through contact inhibition. In density-dependent inhibition, crowded cells stop dividing when the cell surface touches the adjacent cell. So think of a crowded room. In this room that you're sitting in, you have 72 students. If every student created a clone, you have 150 students. If every student then created another clone, you have roughly 300 students. 300 students would not be able to fit into this classroom. So once this room is standing room only, no more division would occur. So that's what cells typically follow, that law. In the case of cancer, these cells no longer obey density-dependent inhibition. So therefore, the 300 students would keep dividing to 600 to 1200 to 2400. So you would keep dividing and you would just pile up on one another, and then you would eventually create what is called a cyst. So that is cancer in a nutshell. It no longer follows density-dependent inhibition. This shows an experiment called cell culturing. What we do is we would put several cells in this type of flask with nutrient media that could be extracted from a dead horse or a dead cow, typically both. We call it fetal bovine serum or horse serum. You put a few antibiotics in it, you put some growth factors in it, and the cells will love it. The cells will divide. They will divide until their cell surface touch the adjacent cell surface. Once that occurs, these cells will stop dividing. In the column to the right, called cancer cells, notice that you no longer have a monolithic layer of cells on the bottom of the flask. These cells are piling up, and they will eventually crowd the flask until they're full. These are the types of cells you want to use in experimentation. You do not want your cell to stop dividing while you are treating the cell with a drug. So we typically use cancerous cells like neuroblastoma. This is a normal mammalian cell that follows the rules of density-dependent inhibition. This is an abnormal cell called a cancer cell. Notice that they are piling up on top of one another. This is not normal.